Hey, business and biotechers, this is Matt Pillar. And before we jump into today's episode, I have a big special announcement. On Monday, November 13th at 11 a.m. Eastern, we're taking the Business of Biotech live for a 1-hour, highly interactive web conversation with a special guest you won't want to miss. I'm planning a good 30-minute grilling of Blue Sphere Bio CEO and biotech legal expert, Keir Loiacono, on legal and IP protection considerations for new and emerging biotechs. Keir is an Esquire and a veteran biotech attorney turned CEO with a ton of experience. And when I'm done with him, I'll turn him over to a select group of viewers, you business of biotechers, who will have the benefit of 30 minutes of face-to-face Q&A and interaction with Keir and other members of the call. This hour will be incredibly consultative and offer great value at no cost but your time. And it's an opportunity for some face-to-face virtual networking with fellow business of biotech listeners who just so happen to be the coolest cats in the business. Go to the link in the show notes of today's episode to register, and I'll see you there. So welcome back to the Business of Biotech. I'm Matt Pillar, and today finds me in Cambridge, Massachusetts at Lab Central, visiting Joe Kaufmann, Dr. Joe Kaufmann at Codagenix, SVP of Oncology? EVP of Oncology. All right, perfect. And Joe, I wanted to have you on the show since you joined me on a panel discussion on therapeutic vaccines a few months ago, because you did a fantastic job on that panel. So ever since then, I've been like, I got to get down there and get some time with Joe. So I appreciate you for having me here at the facility. It's gorgeous. It's a great spot.
Thank you for being here. I look forward to the conversation.
Yeah, me too. So I want to start by getting to know you a little bit better. You earned your Ph.D. in Germany. Where'd you get your Ph.D.?
At German Cancer Research Center, which is essentially the German version of the NCI, if you want to think about it like that. It's associated with University of Heidelberg in southwest of Germany.
Perfect. So you earned your Ph.D. there. And then you came to the U.S. and did your postdoc at Mayo?
At Brigham and Women's Hospital. Mayo would be the other option if you wanted to continue to work on oncolytic viruses. That would have been natural.
Yeah.
But I ended up working with Nino Kioka in the neurosurgery department at Brigham and Women's Hospital.
Very cool. And that's here in Boston?
That's here in Boston, correct.
So what was... Take us back to that day, like those days. What was your mindset? Like, why did you want to come to the U.S.? How did you end up here?
Yeah, I guess I have to disappoint you to say that was a scientific decision. I think it was primarily driven by the fact that during my PhD, I had met an American citizen and who ended up being my husband. Mm-hmm.
Well, that's not scientific, that's love.
That's love, exactly. And about 2 years into that relationship, after he had been in Germany for about 11 years, he decided that it was time for him to move back to the States and asked me if I would want to join him. And when I realized, of course, he's from the Boston area, and that is the location where we would consider moving to. Yeah. Where else to go as a scientist or as an aspiring scientist in this area? And so my condition was I have to graduate and finish my PhD. But ultimately, I think I interviewed at Brigham and Women's during Blizzard Nemo in 2013. A week before my PhD defense, and the rest is history with my postdoc, and we have been here ever since.
Yeah, well, that's a great story. Your husband is from Boston to begin with?
Yeah, from the area.
Yeah, very cool, very cool. What took him to Germany? It's like the opposite question. I know what brought you here. What took him there?
He moved for work as well. He is an IT data storage solutions consultant. Mm-hmm. Initially had a gig 1 week here, 1 week there, and decided that is not sustainable after a while, even as a 25-year-old, and that's how he permanently moved to Germany.
Yeah. Very good. Okay, so why immuno-oncology? Where is your interest there rooted?
Yeah, I think it's more about the oncology part of this that really drove me there. I think I inherited my dad's interest in biology and science in general. He's a biology, chemistry teacher. And while the majority of my upbringing was influenced by sort of my mom's artsy side of things, I really also always enjoyed watching nature shows and things like that. I actually do that to this day. Just last week I learned something wild about some frogs in Patagonia.
Wow.
But that's beside the point. As I'm fascinated by all of that, it always felt like, okay, that's great that we understand how animals interact with each other or symbiosis between different things, but somehow that isn't applied enough for me. And so that's where I think the desire to do something sort of biomedical arose. I then had the opportunity as a teenager to live in New Zealand for a year for a student exchange. I was able to do that through Rotary International and met some fantastic host parents and others who allowed me to have a very influential time, I would say. One of those host families there, my host dad had gone through several rounds of melanoma at the time. And there was always this underlying concern about when would it recur, and if it does, what would be the repercussions of that. And that somehow got me into thinking about oncology. I ran a half marathon for New Zealand Cancer Society as a fundraiser supported by Rotary International. And well, I should say I ran half of that and walked the rest, but but somehow that got me into things, and it was really that passion to do something for cancer patients that drove me into that direction. I think immuno-oncology as a field was still sort of budding and emerging, and there were multiple twists and turns that ultimately got me into immuno-oncology specifically, but oncology is what I'm passionate about to this day, and if anything, as you grow older, it gets closer and closer to you as you realize that it affects your own life and that of your friends and family.
Right, yeah. So, spent time in New Zealand. I wanted to ask you, total aside, just a total aside, but your English is like, it's impeccable.
Oh, thank you.
How did you get so good at English?
I guess New Zealand was part of that. Listening to the accent when I came back and talked Kiwi accent, I went beard. I think I had actually trouble with the German English teachers who couldn't understand me. But then all of my work is in English. Every paper and publication is in English. I speak English at home with my husband and did ever since we met. So it's just
it's fantastic.
Immersive. And part of it is also my approach to assimilation, I guess. If you go to a different country, you live like they do there to the degree that it aligns with your values. But, it doesn't help me to find, a German enclave here and pretend that I live in Germany.
Right. So. Yeah. That's fantastic. So when you were making decisions about how you wanted to apply your passion in oncology, what led you down the path of biopharma, the biopharma industry, developing biopharmaceuticals? Why not academia? Why not practicing medicine?
Yeah.
What was...
on the academic side, I could give you the usual story about academic funding cycles and the politics behind that. I don't think I was close enough to really having to get funding myself so that I make this the main reason. For me, it was really how to apply the science and make an impact for patients. While both my Ph.D. and my postdoc institutions were hospitals or very closely affiliated with hospitals, so you would think that there was translational science going on, the work I did personally and the visibility I had there wasn't close enough to translation and patient impact as I wanted it. So I felt like I needed a different approach to that, and that's how I ended up in biopharma. Now, the question, why wouldn't you do that as a medical doctor? That seems even more intuitive. Again, is, I guess, the more humbling version of that story. After returning from New Zealand, I did another student exchange, this time shorter, and to Argentina, where I had mentioned my ideas about potentially pursuing a career in medical science in some shape or form for oncology. And my host father in Argentina had a friend who was an oncologist, and I was able to shadow this individual for a day to see what it would be like to practice as an oncologist. And let's just say the pediatric cancer patient who got a bedside procedure that I observed did better with that procedure than I did. And I left that day with a giant bruise on my head because I passed out and had to realize that probably practicing medicine, at least at that time in my career, wasn't an option for me, and I wasn't made for it, honestly. And so I'm lucky that I was able to pursue a biomedical career and still try and work towards the same objectives, just in a different way.
Yeah. I mean, I've had plenty of conversations with, asked the same question of many execs in the biopharma space, and they've told me a few similar stories. The actual clinical work was just not going to work out for them, but also that they were inspired to be able to have a one-to-many impact as opposed to a one-to-one impact. So I mean, there's that bright side, right?
That's fair. I would say that there is still obviously immense value to one-on-one care and relationships. I will say also probably at the time I wouldn't have had the interpersonal skills to actually practice, especially in a difficult area like oncology, where it's not only scientifically or medically difficult, but also obviously on a very personal level, very difficult for the patients, their families, and those around them. Yeah.
So what were your early research days like? Like what were you researching early on in your career?
Yeah, I think during my Ph.D. and postdoc, I worked on this modality that I think today we generally call oncolytic viruses. They do a lot more than that. They're actually immunotherapy drugs, which is why I don't necessarily like the term, but that's how most people know about them. Essentially, they are viruses that are repurposed in a way that they can destroy cancer cells and re-alert the immune system to attack a person's cancer. And it is, for me, was a perfect marriage between this fascination for these tiny little viruses that actually are not even alive, but can wreak so much havoc if you use them, or if you think about them in a natural way of causing disease, but could also be repurposed and engineered in so many fascinating ways, and then apply that to the oncology disease area that was passionate for me. So that was where it originally came from. Because of the virus experience that I had gained, I was actually recruited into biotech for vaccine development based on the knowledge about viral engineering, viral life cycles, assays, how to assess where a virus goes or the immune response to a virus, and have been always sort of at that intersect between infectious disease and immuno-oncology, where actually a lot of what we know about the immunology of cancer originates from our knowledge about the immunology to pathogens, viruses specifically.
Yeah. You said you recruited to biotech. Tell me about that.
That was actually really strange. So my postdoc was only about 15 months, I would say. And at the time, I started sort of getting itchy to get out and actually try and make the transition into biotech, although I wasn't at the point yet where I was actively looking for a job in that sense. And usually I don't respond to phone calls from numbers I don't know, but for whatever reason that day I did, and it was a headhunter that was trying to sell me this job about herpes viruses, and that I would be perfect for that, to do vaccine development. And I disagreed because I thought of myself as an oncology researcher, but decided, hey, let's take this as an opportunity to, see what interviewing is even like in industry, because I had no idea, and sort of approached it from a learning side of things, and ended up with that company doing herpes virus vaccine development, originally from an infectious disease perspective, although we did expand that herpes virus program also into oncology with a new Epstein-Barr virus program that we started to develop at Genosha Biosciences, which obviously has dual disease area applications.
Yeah, very interesting. We're going to take a quick timeout. I'm going to make sure that my camera's still working. Quick timeout. Yes. And our sound is still working. Good. All right, it makes me nervous, you know.
That's all right.
I would be very disappointed if we did all this and didn't catch
Had to do it again. Come out the same way.
I come back and visit any time, but so your career from there, working with this company that was doing herpes virus research. What did you go from there to Saro?
Yes, I did.
Yeah.
Yes,
Yeah.
I was with Genosha Biosciences for about 3.5 years, working my way up from scientist 1 to scientist 2, and just the regular trajectory, I guess, that you that you have or can have early on in a career. Admittedly, I was supported by a lot of people who believed in me and allowed me to, expand my reach from doing bench science to also having research associates report to me and sort of start project leadership kind of responsibilities. Yeah. But then after about 3.5 years, I decided that it was time for a change, and I joined Tesaro, which was a virtual company, not virtual in the sense of that we think of now after COVID, but virtual in the sense that they do not have their own labs, and science was operationalized entirely through CROs and academic collaborators. And so it felt like a natural sort of progression also maybe to try and get outside of the day-to-day hands-on lab work. Yeah. And it was a natural transition to oversee science that is conducted by others.
Yeah, and I've got some questions for you about sort of that transition into more of a senior leadership role that I'm going to get to later on. But since you brought it up, at what point in your career, in those research days, scientist 1, scientist 2, at what point did you start thinking about potentially down the road moving into more of an executive position? Was it sort of always in the back of your head, like just the natural progression of things, or?
I guess so. It was that. I can't say that there was a trigger event that made me rethink that I don't want to be in lab. It wasn't a broken flow cytometer on a Saturday that made me suddenly decide I need to do something different. Yeah. But it was just, I think, a natural expansion of understanding more and more what it takes to develop a drug, right? You come from scientific training where people teach you how to run an assay properly, and you build from there, and you go to scientific questions and larger question horizons and planning horizons. So it's actually natural to then think about, okay, what does that take in a company to develop a drug? And there is more than the science itself. There is, how do you make that drug? How do you actually prove that it is okay to put in patients? What does it then take to develop that in patients until hopefully finally at the end some of these are then becoming drugs available on the market? And so I think it was a curiosity about that process that made me then think further and further outside of the scientific experiment, that then I think naturally at some point you realize you can't do it all, and you have to sort of think about, what am I good at, and what am I interested in, and where can I pursue my passion of developing drugs, not just discovering them.
Right. Yeah. And then you got quite I guess a baptism by fire for the flavor of Biopharma when Tesaro was acquired by GSK. Sort of a exposure to the business of biotech, right? Mergers, acquisitions, line
I guess
change.
It was the second round of a baptism. When I was at Genosha Biosciences, we unfortunately had to go through some restructuring, so I had been on the surviving end of layoffs, but we can discuss how both the survivors and those who need to exit a company are affected by an event like this. Yeah. Then the merger acquisition was sort of the other side of it, maybe the happier funnier side of things. But people say if you haven't gotten laid off or got acquired, you haven't been in biotech. So just wait for it.
Right.
And yeah, I guess I'm fortunate enough to have been able to learn from both sides.
Yeah. So tell me a little bit about that. Like the first, so at Genosha, how did sort of that tumultuous time affect you, affect your work, even personally?
Yeah, I would say at Genosha, the writing was on the wall a little bit, in the sense that we were all convinced that our phase 3 ready sort of asset was actually something that really should get patient access and should be developed further. And it was thanks to, I'm going to say, open leadership by the CEO, Chip Clark, at the time, that everyone within the company had some visibility to the difficulties we had at the time to find partners that allowed us to finance that phase 3 program, which in the vaccine space is a very big program with safety database requirements that are enormous. So as a small biotech, we just wouldn't have been able to do it on our own. Obviously, not everyone was privy to the details of negotiations that were had or weren't had, but in general, we knew, if some event doesn't occur until a certain time, it will be tough for us to stay fiscally responsible for the rest of the company. So yes, that moment was hard. It was hard to be a manager at the time, getting dragged out of a team meeting to say, hey, there will be invites. Make sure your team doesn't go directly to lab, but goes to their laptops to see which invite they're in. So, I mean, those things, when you go through them for the first time, is half deer in headlights and half what am I doing as a human being,
right.
And some of your team stays and some of your team has to go. And, that's hard to explain when you actually had no influence over decisions like that. But you go along and you try and be a good human and manager and go through that, I guess.
Yeah. I've had conversations on this topic here in Cambridge and Boston with other biotech execs who have said much the same. It's too small a town to, go through it with a chip on your shoulder. I mean, it seems like there's a lot of transparency. Like it seems like it's pretty open in terms of.
I feel like that's part of how you can do this well. Transparency to the appropriate levels, of course, because it will help people understand that it's not about them as a person, which is why they were on the restructured end of things. It wasn't that they are the worst and therefore let go, but here is what we're trying to preserve. Here is what we had to give up. And yes, it was hard for us, but we had to do this. And I feel like if you explain it to people, that doesn't take the emotion on the day away, but in hindsight, I think it helps them understand and not take it too personally and too seriously. And I was very pleased to see that within a few months, pretty much everyone at Genosha had a new role.
Yeah. It's a great place to be able to walk down the hall or down the street, right?
Yeah, exactly.
And market yourself.
Exactly. So then a merger and acquisition is obviously a very different thing. I would say on that front, I did not see that coming, personally, the role that I had at the time.
What role were you in when that?
I think I was somewhere between principal scientist, associate director, which is a lateral part depending on which sort of track you're in. I did not have the visibility that this would be coming at the time, but I will also say that we had quite some time to process this in the way from the original announcement to closure of that, and then officially becoming GSK employee. For me, it was about a year. So with a lot of influential conversations that made that happen. But again, I think it's about how do you message the reasoning. The fact that someone like the CEO of GSK was there on that day, not in her usual orange outfit, but in her Tesaro blue outfit, to be vulnerable and say how nervous she is about making this work and how important we are to make this work and work together, made it human again. And I will say that I don't think Tesaro would have been able to develop the drugs in the way that we were then able to with the support of the GSK team.
Yeah. What kind of change to your day-to-day did that acquisition bring?
For me, it was, let's be real, a massive career accelerator because my day-to-day changed from I'm one of the scientists overseeing some studies and reporting them through a sub-team to a project team who then goes through the layers of decision-making to essentially building out an immune biology group in the Boston area. Obviously, pharma operationalized their science internally to a large extent. So part of this was create an immuno-oncology footprint in Boston. I was able to join the senior leadership team of the immuno-oncology and combinations research unit at GSK with the most incredible human beings I've ever met on a scientific level and as friends and mentors. I learned so much from them scientifically and otherwise, leadership-wise, that I could not have ever imagined. I will also say at Tesaro, immuno-oncology research was actually fairly small, while the company was about 700 employees total at the time. Mm-hmm. Immuno-oncology R&D was very small. So for me, I finally had people who knew what I was talking about and who understood my struggles with an asset or an assay or a certain set of biology or the sort of bigger issue around immunology, where if you tweak in one place, you're actually getting a lot of other reactions that come downstream of that may or may not negate what you're actually trying to do. Right. And those complexities, I think I finally have people to talk to and to learn from and to solve problems with.
When you're part of a small group and you're being acquired by a larger company, common wisdom might you might look at that and you might say, that's sort of a risky place to be. But clearly, you were doing something right. You did something right. If you had to give some advice to folks who find themselves in a similar situation, set your humility on a shelf and tell me what Joe Kaufmann did right to set yourself up for that, as you said, a career acceleration.
Yeah. I would say the main contributor to leveraging the opportunity of the acquisition was to put aside preconceived notions of what pharma would be like. I had been in biotech before, and through friends and otherwise, you hear all these things about politics and layers of processes and just being very corporate. And I allowed myself to just step away from that and just walk the experience and learn from the people who I meet, whether this is a group that I would be willing to work with. I had the chance to very early on meet very influential immuno-oncology peers and colleagues at the end. It was people like Ken Hans, who ended up being one of the other members of the senior leadership team at immuno-oncology, my future manager, Jim Smothers, the head of immuno-oncology, who made an effort to get to know me on the one hand, but on the other hand, I was immediately struck that I would be able to see myself working with them, and that they weren't that picture that I had of what pharma or corporate people would be like, and I allowed myself to experience that. I would also say on the flip side of that I did put conscious effort into the idea of building these relationships, and when I would have been at a conference in New York, I decided that Pennsylvania isn't far away and let me go to Collegeville and meet these people. Mm-hmm. It was actually a unique opportunity to even meet some of the UK colleagues, Sue Griffin, that I ended up working very closely with because she was there at the same time. And it was just a fantastic opportunity to build the relationships and make it more about a personal work environment and not so much about pharma versus biotech and David versus Goliath.
Yeah. What position were you in at GSK when you left?
I was a scientific director at the time, and I was a member of this sort of core leadership team at the research unit. At that time, I had built the immune biology team, at least to some degree, in the Boston area. Had set up a successor to replace me in some shape or form, who's doing a fantastic job from what I hear. I think that's where my position was. I have to say, the acquisition itself and the transition and merging portfolio, data systems, all that stuff took quite some time. So I think I left relatively shortly after we had achieved some sort of steady state, I would say.
Yeah. Was that system, so I mean, the discipline, it seems like the discipline that you were working in with the GSK acquisition, that was sort of supercharged.
Yeah.
What was challenging for you about it? Was it sort of the back-end systems integration, some of that kind of day-to-day, like merger of systems and operations, or?
Yeah, I mean, as you can imagine, if you go through the last 10 years of your email inbox and you try and tell me how I should navigate that inbox, that's probably hard.
Yeah.
And it requires a lot of historical knowledge that honestly I didn't necessarily have. So some of it was me figuring out who 5 years before me someone thought about how to structure files and things like that. But honestly, that's just one of the technical things that are necessary and very important to ultimately creating a properly integrated team and company. I will say, it was not just data integration, it's just, I think, a lot of lessons about scientific rigor, about data robustness, about reporting that are very important and really well intended, can also become a burden if your mindset is that you have to ensure that 100,000 people go in the same direction, at least hopefully the same direction, right? So I understand why a lot of these processes and policies are in place. I will say that I had the benefit, I would say, from getting a lot of career support and development support from my manager and others in the organization, in particular as a female leader. There was a lot of emphasis on inclusion and supporting female leaders. I was admitted to a program called Accelerating Differences that got me access to personal coaching, group coaching, learning about yourself and who you are as a person and as a leader. It was opportunities like this that were the good end of all these offerings and things. On the other side, to create visibility, I was brought into a GSK-wide initiative, how we should all be writing, our electronic notebooks the identical way. And it was at some point taking me a little bit too far away from the science and the patient-applied translational work that I was passionate about. That ultimately, despite the most amazing team I could have thought of, made me consider other options.
For emerging biotechs, scaling the process development and manufacturing of biologic molecules to clinical standards can be challenging. However, you don't need to go it alone. Don't miss an episode of the Business of Biotech podcast, where we offer insights on regulatory, funding, and other essential topics. The pod is brought to you in collaboration with Cytiva, a global provider of technologies and services that advance and accelerate the development, manufacture, and delivery of therapeutics. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech.
All right, so one more question about that before we transition to the other options that you considered. You mentioned the programs that you were exposed to that helped you develop your career and your leadership skills. And that's fantastic, because I think a lot of, you hate to stereotype, but I think a lot of folks who start out in research as research scientists maybe have a bit of a difficult time, a challenge transitioning from, lab work and sort of being in your own head and doing your work to opportunities beyond that encourage leadership. So what advice would you have for folks who maybe don't have inherently, I mean, you're a super personable person. I imagine you were that way before you took all these classes, but if it doesn't come naturally to you, what advice would you have for research scientists who are interested in sort of climbing out of their shell and pursuing other options?
Thanks for the kind words. The reality is it does not come natural.
You fooled me.
And it is a lot of work, I think, to first of all learn who you are and what you're about, because you cannot go out and stand up for your values or your ideas or claim that you can represent a team in an external representation way just as much as in a day-to-day situation if you don't know what you're actually trying to do. So in a way, these courses allowed me to take time to reflect on why am I the way I am? Why do I have reactions to certain things the way I do, the good, the bad, the ugly, and you have to put in the self-work to realize, okay, I'm getting angry with this person in a meeting because now for the third time they were late. Part of that is because I'm German and I was trained to be on time all the time, but what it's really about is respect, and I want to be respected. I try and respect others, and that's why I get annoyed. So either I have to explain to people, please be on time, or you find your own way of then how to handle these kinds of situations. Just say, okay, they are showing respect in a different way. That's also fine, as long as otherwise I feel respected. Yeah. I mean, this is a tiny example, but I think you have to put in self-work. If someone wants advice on what books to read, just go to BrenéBrown.com and you will find all my secret sauces.
Brené Brown, I duly noted.
That's where I learned to use her words, dare to lead, I guess, and also brave the wilderness. I'm using these stereotypical book titles of hers, but it encompasses in a way what you have to go through. And for me, it started very self-reflective, and only because of that, I then found my way to show up in a certain way. That doesn't mean when we leave the room, I need 5 minutes for myself to de-stress. But maybe that's part of the skill as well, to show up in a certain way, in the right moment, in the right way.
I appreciate that, the transparency in that response. And at risk of creating another stereotype, my wife is 100% German, so I know well the expectations, right?
You were 20 minutes early here, so I appreciate it.
I've been married for 20 years to a German woman. I know the deal. Okay, so that transition to Codagenix. Yeah. You hinted to the why a few minutes ago. You talked about the maybe desire to get closer to. I
think it was on the one hand, the growing desire to get back to a more innovative, faster, nimble way of operationalizing science. It was also, I think, the natural transition because of all that leadership support that I had gotten. That when I was approached about an opportunity to build a disease area from scratch, to take a platform technology and apply it in a different way than what Codagenix had been done previously, going back to science I was very familiar with these oncolytic viruses. In a way, it's a full circle kind of situation here. That sounded very attractive to me at the time and allowed me to put to the test whether I was made out for this and figure out what it would be like to apply everything that I had learned in part theoretically, but in part maybe also practically, and see where this would go.
Yeah. Was there any reservation or concern about, you would obviously sort of rode that acquisition train to big bio, and now you're coming back down to a small clinical stage, or early stage bio. Was there any reservation about working for a new and emerging company?
No, I think I understand by now enough the cycles that things go through. I mean, obviously, what we have experienced in a macro climate in the last couple of years or so is unique, but cycles similar to this, I would say, have existed before, and in a way, Cambridge or the Boston area allows you to be free in a way because there are so many other local opportunities that you could pursue in the event that, end up on the restructured end of difficult decisions that leaders would have to make. So in a way, this is par for the course. And if anything, my reservations about this transition was, is it really worth leaving such an amazing leadership team and peer team behind?
Yeah.
Is it worth giving that up for the format of how a company operates?
Mm-hmm. Speaking of the format of how a company operates, what adjustments did you find yourself having to make? And again, I fully recognize that you would, before the GSK Tesaro experience, you had seen it before, but it had been a little while. So, what adjustments did you have to make professionally or accept, in terms of resources that are available to you? Or, it's obviously a pretty big transition.
Yeah. Well, let's be real, just because a company like GSK has a big purse in its entirety, that doesn't mean that within your research unit or work business unit, you don't have to fight for resources and explain why it's worth investing them here and not somewhere else. So I did not experience, at pharma you just, throw the money out there and just blank check, do whatever you want. Yeah. And then suddenly you find yourself in biotech where you count your pennies. I don't think that is a fair representation. How thoughtfully, at least GSK in my experience, tried to spend their money. It's more about, you go from very well-organized systems. I always said that I had a tracker to make sure I don't forget about all the other trackers I need to fill out. You go from that to more or less build the plane as you go along, as you're maturing as a biotech company. That gives a lot of opportunity for influencing what this company should look like, but at the same time, you sometimes just have to find a way and troubleshoot, I guess, your day-to-day work as much as you troubleshoot your science.
Do you like that? Do you find that exciting?
Yeah, I think if I am in both extremes, it's no good. I think my sweet spot would be somewhere in the middle. But if I had to make the choice, I would rather have a little bit more freedom and allow to make decisions faster and quicker, and then act on them rather than going through several layers of approval.
Yeah. All right, you better check that. I'm sorry, I have to keep stopping with it. Still good. I think it's stopping at the 30-minute mark, so I'm going to check it again in about 10.
Okay.
So when you joined Codagenix, what position did you take there?
I was hired as EVP of Oncology.
Oh, you were, okay.
At Codagenix.
Yeah, and was that, so you mentioned obviously getting closer to development and patient benefit as being a motivator, but was the EVP opportunity enticing to you as well? Was that?
Well, I think it was pretty scary, and when I was first approached about a position with that title, I said I have no business applying for this or ever being considered for this. I didn't see myself being ready for that. Again, at the height of biotech, let's just put that out there, titles is letters on a piece of paper, and it's about what you contribute and not what your title is.
Yeah.
So for me, the job description of the idea, first of all, to build a lab, find lab in a new location. So we are here in Cambridge, as you mentioned in the very beginning. Codagenix is headquartered in Farmingdale, New York on Long Island, and that's where our vaccine R&D is located. But we had the intention to build the oncology research and development team in the Boston area, but we had no place. So part of it was find a location, build up a operating lab, and also a team. Therefore,
so this fell under, this was your responsibility?
Yep, that was part of the gig. Mm-hmm. At the same time, obviously we had proof of concept data with a promising asset that was studied to the degree that made people confident enough that we should be building this disease area, which was new for Codagenix, but basically take that towards an IND or clinical development and think about how do we build a portfolio behind that. So I think it was the broad scope to think about discovery to translational early stage clinical and build it in a new location from the ground up with an ability to therefore also influence team culture and hopefully therefore company culture was very exciting for me as an opportunity, letters aside.
I mean, letters aside, you just rattled off a whole lot of responsibility, Joe. I mean, you know, find a location, build a team, you know, handle that discovery through translational, prepare for IND. Like that, this is a lot. Is that why initially you're kind of like, I don't know if I have any, like, what was your reservation?
My reservation was honestly, and I shouldn't be saying this as a trained female leader, that it was the gut reaction to those letters or that title. It wasn't the scope. The scope was actually
achievable, like in your mind, I can go do that.
Hopefully achievable. It was like, this sounds pretty much like exactly what I want to do, so let me see if I'm ready for it.
Yeah.
Honestly.
Yeah.
Challenge. Challenge and opportunity, it always comes together.
Yeah.
Yeah.
What did you have to work with when you took the position? Like what was here? You mentioned that your headquarters are in Farmingdale.
Yeah, so I had one scientist reporting to me in Farmingdale who continued to conduct mechanism of action studies to support an IND for that lead asset. Mm-hmm. I obviously had cross-functional support from regulatory and medical and CMC and other departments that to that date primarily were focused on infectious disease assets and live attenuated vaccines. But I did have, that expertise to help with. I worked from home in the beginning until we went through a whole bunch of application processes for lab space at the time. Around that time, the real estate market in the Boston area was really tough. It was almost impossible to find anything, so we were very grateful that we had the opportunity to be accepted to one of the Lab Central locations here and tap into that community of biotechs, emerging biotechs that Lab Central creates really well together with their partners and sponsors. So eventually we moved into the offices, then our labs became operational about a little over a year ago now. So it's a process.
Yeah. What are some of the, just curious, what some of the criteria are? You said you were fortunate to be accepted into the Lab Central space. Are there certain criteria that Lab Central looks for in a tenant?
Yes,
detail.
You obviously have to have an interesting plan for growth that they, with their respective site sponsors, are interested in. Mm-hmm. Frankly, there are some criteria related to not being a direct competitor to another resident company. Right. That is out of your control in a way. Luckily, Lab Central does not have super strict restrictions on dollars amounts raised at the time of application, which is in contrast to some of the other incubators that are really focused on seed stage companies, while Lab Central is focused on helping biotech companies at all stages. Yes, their original location is supporting 3 scientists at a shared bench somewhere. But by the time you get to some of the other locations, they allow you to be a more mature and larger organization. So they are less strict on funds in that sense, or fundraise history, I should say, which made it easier for us to be admitted, in the sense that while we were pitching for a relatively small team in this new disease area, the company had been in existence for 10 years and is much more established in the infectious disease side. So in a way, we're building a startup within a biotech, and we needed to communicate that appropriately, why we fit into an incubator-related space like this.
Yeah. So the transition from some of the hands-on, and I know it happened gradually over time, but now, to your point, you find yourself in an EVP role, building infrastructure and people and teams. Do you miss the research? Do you miss the science? Or are you still involved? Tell me a little bit about.
So I don't miss being in lab. Okay. Very clear for me that I feel more passionate about how I can influence science from a non-bench-based position. But I am still deeply involved in the science. It's actually part of the beauty of this role that I have, that I can sit in one meeting describing how to analyze an ELISA assay and how to apply that, and how we can graph something complex in an intuitive way. And in the next meeting, you talk about staffing or budgets or whatever it is. So it's the diversity of that is actually appealing, and I don't consider myself removed from the science. In fact, if anything, that is something I would advise people to stay true to the scientific depth that you need to have to actually be well versed in what you're trying to accomplish. It is easy to go broad and do all these shiny things. You get to go to present, you get to do this kind of interview, and that's all nice, but at the end of the day, your job is to develop drugs for patients, and you do that through deep scientific knowledge. If you're coming from the science track, obviously, if you have other types of responsibilities, different roles, it comes from a different angle, but you have to have a technical expertise, and you need to find a way to stay deep enough on that to hold your ground, honestly.
How do you maintain that? What are some of the things you do daily and are you plugged in with your lab employees?
It's part of, but that is more helping the science you create afterwards, right? I think for me, it's honestly PubMed alerts. It's my favorite thing. Every morning I open my inbox, and while it's deer in headlights of the number of emails, many of them are actually my PubMed alerts that push me titles and abstracts, which at least allows me to monitor trends, what is hip and what isn't. If there are new words that then keep coming up all the time in titles, then obviously there's something to that biology that I should be aware of, and it helps you stay on top of it. I am very fortunate that I get to go to a lot of scientific conferences, where I also get to, stay on top of what's going on, and sometimes, again, follow trends. And I do try and put placeholders in my calendar for personal thinking and reading and things like that. And yes, those are the slots that get pushed around relatively easily, unfortunately. But at least every week I have a reminder in my calendar that tells me that I should be focusing on this, and you try your best.
Yeah. What are some of your favorite resources? You mentioned scientific conferences. What are some of your favorite resources to stay up on scientific trends?
It's conferences, I actually really like panel discussions and fireside chats these days. I always dismiss that as something that isn't deep enough, but now I actually really appreciate hearing from very senior people thinking about the field and the trends as a whole. So there were also consulting companies that put these things out in webinars. They're a platform like yours where experts get to talk about their science, and it's deep enough that if there's something that's connected to what you're trying to do, or something that you're really curious about that's new to you, that you have enough tools and names to look and then do a little bit of your own research. Yeah. I think it's I never appreciated how much quote free content there is out there actually, and how much people are actually willing to share in some of these interviews. Obviously not their secret sauce, but their general thoughts around where the field should go, and particularly in immuno-oncology that's important because, let's be real, despite billions of dollars having been invested in immuno-oncology, with some incremental improvements over what the PD-1, PD-L1 axis inhibitors can do, we haven't made the next big leap. Yeah. And so I think putting all of our knowledge and minds together to do the next big improvement and transformation for patients, I think, it's probably not going to be solved by one individual.
Right. Yeah. I mean, those panels you mentioned, you were, as I mentioned, you were my guest on one. I believe it was back in May, and did a fantastic job there. And I know it's been a short time, right? We're looking at the fall. So it hasn't been a ton of time, but I wanted to get an update on what's going on at Codagenix since we last spoke. And to your point, I'm not expecting you to spill the secret sauce, but you share with me some of the work that you're doing now.
Yeah, thanks. On the oncology side, I think we have been making progress on both deeper understanding of how our lead asset, which is an influenza-based oncolytic virus targeted for breast cancer immunotherapy, how that works in particular in the context of standard of care therapies in the breast cancer space, which is continuous work we have been incrementally building on over the last few years. And we've made some progress also on our discovery efforts and portfolio build efforts to be shared at a future date when mature enough. On the other side at Codagenix, on the infectious disease side, obviously we have several ongoing clinical trials in a variety of spaces, and we are working hard to move those along. And again, we'll release the appropriate data when those clinical data sets are ready.
What is next for you in terms of the mission that you accepted when you came to Codagenix? Like you rattled off all these, all these goals, all these responsibilities. Obviously, you've made great progress. What boxes still need to be checked off, or what are some of the forthcoming boxes for Dr. Joe Kaufmann?
Well, I think what I would like to focus on even more so as we hopefully accelerate out of 2023 into the next fundraising round and things like that is, I think, apart from staying on track scientifically and portfolio-wise, is to continue to build a team culture that really ensures we are a high-performing team, and hopefully, test run some of those initiatives that support scientific development of staff in a way here in Cambridge. But obviously, the idea is to apply those things and make them, in parallel to similar initiatives in other parts of the organization. I think we need to think more about how do we help our scientists do the best job that they can do. And some of that is, giving back to my team members what other leaders did for me, and give them opportunities and help them go out there and learn and talk about their science and get
Yeah.
the tough questions and go through the motion of maybe not having an answer today, but then thinking about it and being better prepared and seeing their own progression about their scientific knowledge, about how they apply that scientific knowledge, and how that fits into the larger development process as a whole.
Yeah. How big is the team here in Cambridge?
Our oncology R&D team has currently 3 bench-based scientists, and given that there is a process development focus at this particular lab location or lab central location, we are also building our process development team here, which is soon to be 2.
Oh, very good. Very cool. Good stuff. The labs are right here in this,
Yep.
in this facility?
Yes, there are. Yep,
Outstanding.
across the bridge.
Across the bridge, all right. Is it, as far as like team growth is concerned, are you do you leverage the great academic institutions here in Boston and Cambridge? Is that kind of where your core
I
comes
think,
from?
well, we always are supposed to be saying, yes, of course, we are leveraging this.
Right.
And you do leverage it in the sense of the talent pool here in this area is obviously incredible because of all these fantastic institutions that are a short walk away from here. Again, there is a lot of core facility access and things like that through the Lab Central or the larger mass bio network we are able to tap into. If you're asking me, are you constantly going to seminars down the road? I mean, that is always the dream that you would have time for that. But I sometimes wonder if we should be doing more of that actually and really tapping into all the different opportunities we have by being here.
Yeah, well, I mean, that would be more than a full-time job in and of itself.
Yeah.
What parting advice would you give young scientific professionals who sort of want to plot the same course that you did and continue to do?
I would say stay open to opportunities. Maybe what has come through those conversations, some of the influential things I look back on in my career are actually not things I planned for. So it is about seeking opportunities and making the best out of it. At the same time, I think it's really important to maintain a network and relationships. I know that seems to be a beaten horse sometimes, and people always refer to their network and are proud of the connections they have on LinkedIn by the numbers. I don't mean superficial things. I mean actually true connection to people, and to staying open to their points of view that are different and maybe challenge you sometimes.
Yeah.
I think that's the key.
Well, that's fantastic. I appreciate that. And I appreciate you for allowing me to come into the office today and spend some time with you. I've enjoyed it. I think you did a great job and offered some terrific insight for our audience. And I look forward to maintaining my place in your network and hopefully working with you again sometime
Thank you.
soon.
Thank you so much for being here.
Thank
I
you,
appreciate
Joe.
it.
Thank you. So that's Codagenix EVP of Oncology, Dr. Johanna Kaufmann. I'm Matt Pillar. This is The Business of Biotech. We're produced by Life Science Connect in partnership with Cytiva. Check us out at bioprocessonline.com/BOB. And thanks for listening.